So with that, I'll ask Ruedi to come up, who is, I guess you probably know who he is, the head of the group here, and has been very involved in our group. This group in Zurich has been very involved in technical developments in DIA and SWATH, and he's going to give us some considerations about how to use SWATH in experimental biology. So.
Okay, good morning. It's a pleasure to see so many students here. This is actually, the class is about twice as big, so which is deliberate, because we thought the actual instructors thought they could handle a larger class, and there clearly is a demand, so we'll see how it works out with such a big group, but I'm pretty sure that it will work out fine. I would like to also thank the organizers of this course. So we've had, this is actually roughly the second generation of instructors. This course was remarkably entirely conceived and set up and also organized by students and postdocs in the group. Many of these have now moved on, and the present lab members have taken over and have expanded the course and changed its focus. So I think it's a really nice example of teamwork that works, that runs very well. So what I would like to do this morning is to make some comments about the use of DIA, mass spectrometry, in experimental biology. Because I think we not really, at least in our group, we're not trying to develop techniques for the technique's sake, let's say to have a new world record of number of proteins identified or the highest sensitivity achieved. But what we try to do is to come up with techniques which allow us to address new biological questions or old biological questions in a new way. And so it turns out that this development of new techniques is necessary in biology, because a lot of the questions that we would like to answer have been kind of off target yet with existing techniques. So this is basically what I'd like to discuss this morning, how this technique was driven by biological question, how now it can start to make an impact into some research directions. So first I make some general introductory statements. The first is that we focus this course almost completely on data analysis. And this turns out that even just before DIA, the data analysis part of a proteomic experiment has been the Achilles heel. So this has been turned out to be the most complicated component of an experiment where most experiments and most studies fail. And this is actually remarkable, because when you go to a conference like ASMS, you always hear 90% plus probably of talks about instrumentation, about tuning instruments, and about getting samples processed. But this is a graph that came out of an old study. It was in 2009, so it's almost approaching 10 years, which was a very simple study in the world of data-dependent analysis, shotgun sequencing. And a group around John Bergeron put together a sample of 20 highly purified proteins at equimolar levels. So this seems to be a very simple analytical task for a mass spectrometrist to find the right proteins in this sample. And then they sent this sample around to 27 groups who agreed to participate, and they then wanted to see what came out. And actually, the results were pretty abysmal. I don't want to go through that. This has been published a while back. And then we suggested that one would, rather than say, well, it didn't really work, one would try to find out why did it not work. And then the authors, including also the editor of the journal where this eventually appeared, they agreed that the aggregate data, the raw data as they came off the instrument, would be sent to a group, and this happened to be the group of Eric Deutsch at the Institute for Systems Biology, who had established a pipeline to analyze such data, to see how many proteins a consistent data analysis pipeline would find. And the results were that when we break down into the overall process into sample handling, complexity reduction, basically some kind of fractionation, chromatography, mass spectrometry, and informatics data analysis. By far the biggest source of failure of finding the proteins was not the mass spectrometer, was not the sample, it was the informatics. So I think this has even been accentuated in world of DIA, because the actual data generation is relatively straightforward. I think actually quite straightforward, and a lot of the emphasis has shifted onto the data analysis, and that's why we would want to discuss this here in some detail. So the second comment I would like to make as kind of introductory comment is one about general trends in biological experimentation. So here, this is a slide that came out in the year 2000 when President Clinton and Prime Minister Blair at the time announced the draft of the human genome sequence. Of course, they didn't actually do it themselves, but they had the pleasure to do the announcement. But with this announcement came specific comments or expectations, and they all are related to medical questions, but they said one would be able to alert patients that they're at risk for diseases, so this is kind of a biomarker issue, predict the course of disease, diagnose disease, find the best treatment, and so on. So these were very explicit and very important predictions, and then they really were not really fulfilled. I mean, the expectation here was that by looking at a database, which is basically the linear sequence of the human genome, one would learn enough biology to make such statements. And even today, this has not really happened. I mean, no one can actually take a human genome sequence and, let's say, artificially introduce a mutation in a particular locus and predict what the effect of this locus would be, for instance, for a person. So, but what happened after is, so this was roughly when this genome sequence was announced, we saw, of course, and everyone probably knows that and has certain experience that maybe in their own labs, this enormous speed up and decay in price in genome sequencing. So what the first genome sequence achieved was being a landmark accomplishment, and it cost billions of dollars to generate. And now, here, I mean, this is a slide from NHGRI, but now we are further out, and this has been continued to drop. And so now everyone who is, even a graduate student who is in the laboratory, they can go and say, reasonably propose that he would want to sequence a number of genomes in for instance, a PhD thesis for some purpose. So I think what we learned from there, genomics, is that the fast and accurate and highly reproducible measurement of thousands of genomes in the context of GWAS, transcriptomes, exome sequencing, and whatever, I mean, there's a whole number of flavors of genomics, of course, has really transformed biology. So it wasn't the initial database, although this was a monumental achievement, but it was the ability for a large number of researchers to go and do quite precise and accessible measurements that really has had a tremendous effect. So what about proteomics? So in proteomics, I think we're always tracking behind, to some extent technically and conceptually, to genomics. But we now also can say that we have roughly a draft of the human proteome available, and the draft is not complete, but it is quite reliable. So this is a slide that is again from Eric Deutsch, who was running a program which is referred to as the Human Peptide Atlas. And there's actually several of such repositories. I'm just using the one because this is the one we started a while back. But there's also similar repositories from Mike McCoss's group, from EBI at Pride, and from Ron Beavis. So, but they all in one way or another attempt to collect data from the community, mass spec data, and represent this data back to the community for some purpose. The purposes are somewhat different, but that's not the topic here. So in any case, what Eric started doing in early 2000, like 2002 or 3 when this project started, is we started to collect data, and we basically asked scientists who do the DDA, shotgun experiments, to donate their raw spectra. And this is initially was slow, but then we saw this really massive increase, and now there's multiple tens of terabytes of data have been generated and donated. And then Eric and his team, they analyzed this data through a pipeline. It's called the transproteomic pipeline, but the other tools could also be used. So the issue is that this pipeline has a fairly sophisticated way to control false positive identifications, both at the peptide and at the protein level. So we believe that the data that are coming out, and they're represented, for instance, here at this graph, are roughly accurate. And the reason we can state that is because several other people, like also the group from Matthias Mann, and also the group from Alexei Neshvisky, who will be here, they analyzed roughly the same data. The data are freely shared and come to similar conclusions. So what are the conclusions? In terms of having a draft representation of the human genome, there are now roughly 1 million spectra, 1 million peptides have been identified. And this translates into about slightly more than 14,000 proteins or open reading frames or loci that have been touched by these proteins. So we see here there's a couple of points which are interesting. One is that even though very large data sets, and these are very large data sets which are donated, are added to the already existing ensemble of hundreds of millions of spectra, these data sets do not uncover a large number of new peptides and virtually no additional proteins. So what we could conclude here is by the techniques that we have available to explore the proteome, the equivalent of doing the initial genome map, is we reached roughly saturation, and this saturation is nowhere near the complete coverage of the proteome. So what we would conclude is that if we is basically several things. The first is there's still a fair amount of proteins to be discovered if we assume that the human genome contains roughly close to 20,000 ORFs. Second conclusion would be that if we continue just throwing the same technique at the proteome, we will unlikely cover all the missing proteins. And third conclusion would be that we would likely to already be able to discover a lot of new biology if we were to operate and explore the information in these proteins here. So personally, I think we don't need, although it would be nice, but I don't think we don't need to know every last protein that conceivably can be expressed in the human cell if we want to carry out biological experimentation. This is already an extremely rich source of information. So what we concluded when we saw these trends some years back, we concluded that eventually this saturation would happen and that we would like to, exactly as happened in genomics, we would like to come up with a strategy, or it would be useful for experimental biology to generate a strategy where we could do fast, accurate, and highly reproducible quantification of thousands of proteins, ideally all the proteins. But we cannot do all the proteins, but we thought that it would be merit, that if we could achieve these goals here, then we could learn a lot of new biology. And this is really the general idea behind these DIA or SWATH techniques, that they allow a laboratory, even a laboratory that's not really equipped with huge numbers of mass spectrometers, to generate highly reproducible protein maps and of many samples with relatively moderate experimental input. And then, of course, the data analysis is another question that we're going to discuss this week. Okay, so we proposed at the time, this is a few years back, that eventually we would have a complete or at least saturated proteome map by MS. And this is, of course, achieved by discovery proteomics, and this is continued. So there is specific effort ongoing, a coordinated effort by the Human Proteome Organization to find these roughly 4,000 missing proteins. But we thought that even now we would be able to use the present information, and we would use the spectra from discovery proteomics, which basically highlight which peptides a protein in the human proteome produces, how it fragments, where it dilutes, under what conditions it's generated, and so on. And then we would use this information from these maps, basically as prior information, to do rapid quantification of as many proteins as possible. So this is kind of the original idea, and where we would place that. So it's basically, we view it as not a mass spectrometric development per se, but a tool to do new biology. Okay, so now what I'm going to get on in the next few minutes is a few points. This is a menu of points, and I don't think, maybe I won't get to this last point, but we'll see how times will go. So the first point I would like to make is that the data matrix, this would be proteins quantified versus samples or across many samples, is an emerging currency of proteomic experimentation. So now I would like to explain what this matrix means and why we think this is necessary for biological research. So this is a pretty stark deviation to what proteomics has focused mostly on in the past, and that was to identify as many proteins as possible, ideally all the proteins in a sample, or in very few samples. So here we would like to have many samples and get proteins very consistently quantified. So this is what I'm talking about as a matrix. So why do we think this is biologically important? So if you go to a typical biology department, including ours, you will see groups that work in genetics, cell biology, biochemistry, structural biology, and so on. And the general assumption that most of the work in these groups use is that proteins carry out a specific function, and if we understand this if we have the protein identified that carries out a function, we would know the function, or if we have a function, we could purify the protein that carries out that function. For instance, a metabolic enzyme, if we could assay a biochemical function in a metabolic enzymes, if we purify this enzyme, then we have a link between function and the molecular entity that carries it out. So for that, discover proteomics has been the technique that has been used. And of course, there it makes sense that if we want to discover a protein that carries out a specific function, then we need to sequence and identify as many proteins as possible. Because if we only sequence 1,000 proteins, it is very likely that from a sample, it's very likely that the protein we actually seek is not in these 1,000 proteins, and therefore we'll never find it. So the main technique that has been used in these research groups, and in fact by far most predominantly still in proteomics, is to ask the question: can we discover the protein that carries out a specific function? We need to discover as many proteins as possible, ideally all to have a chance to find the protein that carries out that function, and it is clearly is the technique that does that best is data-dependent analysis, shotgun sequencing, and so on. So now, this of course leads to a problem that we all were encountering. Let's say you were analyzing by two proteomes, for instance, a proteome from a resting cell or from a healthy donor and from a proteome from a tumor. And if you'd like to find differences between these two samples, which is a very typical workflow in proteomics, then we're overwhelmed with information. Because when you would use the quantitative DDA techniques, you will now, these days, with some skill, maybe identify from such a sample 6, 7, 8,000 proteins, of which usually about 10% is up or down regulated, almost irrespectively what one does to the cell. So one is sitting there with a very long list of proteins that clearly change between two conditions. But we don't know which one of these is actually important, whether one specifically is important, and how to find that. So this has been one of the big problems from the very successful DDA techniques. They almost uncover too much information. And then the biologists, of course, can pick one out or several and send a student on its way to say, figure out how this change that is observed between, let's say, two states tells us something biologically important. But of course, if the student picks out of 1,000 proteins changing the wrong one, he might never really reach any interesting results. So more recently, specifically also driven by the successes of genomics, basically a different approach to biological research has emerged. And that is the focus on large data sets, also frequently referred to as big data biology. And this is certainly represented by genomics, transcriptomics, increasingly proteomics, and so on. So here, the question is a different one. We would not want to identify which protein is doing exactly what, but we would like to observe what role a protein has in the context of a process. So when this process usually is related or represented as a network of interacting molecules, in this case, this might be, in our case of proteomics, this might be proteins. And we will no longer say this protein is, for instance, doing a specific function. We're no longer saying this protein is an oncogene or is a growth factor, but we will say this protein operates in a context of many proteins which constitute a system. And we want to understand what happens to the system overall if, for instance, this protein is mutated or missing or overexpressed. So rather than trying to reduce the question to what is a specific protein doing, we would rather ask the question, how is the system responding to the change in a protein by sequence or by abundance? So we would learn the behavior of a system from observing it in many conditions. And this brings us exactly back to the issue of genomics that I showed, that the big data world entered genomics by the ability to measure rapidly many genomes or transcriptomes, and I think this now also happens in proteomics through the techniques we describe here. So while we are no longer trying to reduce the activity to a specific protein, but we like to make assumptions about the system, we use also different tools. We would not necessarily use a biochemical tool of purifying a protein and then assaying its function, for instance, determine the catalytic constant of a metabolic enzyme, but we would rather work with statistical association. So we basically have two variables and we see how they correlate. This has been, for instance, implemented in genomics very broadly by genome-wide association studies, where one variable would be some form of phenotype, maybe the propensity of generating for a person to get cancer or diabetes or whatever other disease, and the other would be large number of markers, measured SNPs in this case, measured across a large population. And out of that come associations. They basically show molecules that associate positively or negatively with a particular phenotype, but they do not really imply a causal relationship. They simply are associations. And I think so certainly that's the kind of the direction we're taking in our department here at ETH, is we would like to find as a department, this is probably not possible for an individual research group, but for a department consisting of different groups, we would like to maintain the classical strength of these structural biology, biochemistry, and cell biology groups. But we would like to somehow find a way to relate the statistical associations that come out from this big data world to the mechanistic processes that have been studied for a long time, and find a mutually beneficial arrangement where big data and mechanistic studies can cooperate. This actually is an interesting question, how this happens best. This is not something we have time to discuss here. But what I do believe is that with the techniques that we're now discussing this week, is we can bring proteomics to a level where such associations can be generated, and then an interface can be built between the large data, basically as the statistical world, and the mechanistic world of biological processes. How exactly this happens is interesting research, and it would be interesting to discuss this maybe in a break. So if we want to go that way, we need to have this data matrix that I already mentioned, and we will need to quantify proteins, and we will need to quantify them across many samples, not just 2 or 3, as already been very successful in the DIA world, but maybe this dimension should be hundreds of samples, and I'll come back to the discussion of these dimensions in a minute. But more importantly, it is necessary that each data point or each cell in this matrix has a number. So if you have a large number of missing values, then it is not going to be really useful to do correlative analysis, machine learning, or whatever computational tools you would like to use. So the emphasis clearly is on reproducible measurements and accurate quantification of many samples and many proteins. So with that, so here are just a number of examples of such studies which can take advantage of this data matrix. Biomarker studies, of course, genome-wide or proteome-wide association studies, identification of clusters of proteins behaving similarly across conditions that might not have been functionally linked before, network analysis, population-based molecular biology, and many others. So we think this, if we achieve to generate such data matrices and have the tools to learn the new biology from them, then this will be a very powerful entry into many types of biological investigations. So now, very quickly, because this will be expanded, of course, throughout the course, I very quickly want to touch on how we generate such data matrices by mass spectrometry. And then I talk about the dimensions, which one is important, the X or the Y dimension. So this is what we would like to get. We would like to have all proteins that are expressed in a sample and precisely quantified in many conditions, maybe hundreds. If you're talking about population-based studies, we'll know that from genomics again, that the number of samples to be studied would be in the range of several hundreds to maybe a few thousands. GWAS studies now go to hundreds of thousands of samples, which is probably not achievable for a long time with proteomics, and maybe not necessary. But anyway, this will be certainly in the range of hundreds to, if not thousands, of samples. So, and of course, we show here a matrix, and this should have very few missing values. So we cannot do that yet. I mean, no one really can. First of all, we don't know all the proteins. I showed at the beginning that we saturate out about 14,000 or so, and there's still a fairly large number of proteins to be discovered, which clearly are also biologically relevant. And then, of course, there's the issue of time and resources to do many samples. So all these techniques we're talking about and that are used for the majority of studies today is what's referred to as bottom-up proteomics, and I'm sure everyone is familiar with that. I just want to state the general principle that we actually do not analyze proteins. This would be referred to as top-down proteomics, which is also an interesting approach, of course, but it is not at the same level of robustness as the bottom-up techniques are right now. And we would then measure, we would dissociate this protein into peptides, and the currency of molecules that we actually measure is peptides. And everyone probably knows that. And of course, a number of workflows have been established to separate these peptides, sometimes by 1, 2, or 3 dimensions, and then to generate fragment ion spectra of specific peptides. So what was observed, which was unexpected, is that when we digest a specific protein, any protein, that a very large number of peptides is being generated. So we can, of course, take a protein sequence, and we can put this sequence into a computer program, and we can tell this computer program to cleave the protein apart where the putative protease that we want to use, which is frequently trypsin, would cleave. And then we can ask how many peptides does this protein produce if we were in a computer to digest this protein with trypsin. And the number of these peptides is typically relatively low, maybe in the range of 20 to, of course it depends on the size of the protein, but typically this would be in the range of a few tenths of peptides. In the reality, this is not so. So this was a study that Paola Picotti did here. She was once visiting the lab a while back as a PhD student. And then I asked her to take a number of highly purified proteins, digest them with trypsin, and then measure with what was at the time the best mass spectrometer we had, as many peptides as she could. And it turned out that even relatively short proteins that were highly purified proteins, so the peptides are not from other contaminating proteins, they're all related to this protein, generated in excess of 100 peptides, and this now with the present state of mass spectrometers, I would assume that this would be several hundreds of peptides. Most of them are very, very small ion current, they're very minor products, and they would be things like oxidized, a peptide containing an oxidized methionine, would be peptides that were improperly cleaved, where trypsin did not make a cleavage. I mean, there's all kinds of reasons why so many peptides are being generated. But the fact is, and this has been now many times reobserved, that from a single protein, a very large number of peptides is being generated. So you could say this doesn't really matter, because most of these peptides will be very low intensity in the mass spectrometer. So the signal intensity is very low compared to the major peptides, which tend to be proper tryptic peptides. However, if you have a sample which represents a proteome, where the highest proteins are expressed maybe 6 or 7 orders of magnitude more intensely than the lowest expressed proteins, which happens to be the case in an extract of a human cell, or even worse in blood, then even a minor contaminate or unexpected peptide from a very minor peptide from a highly expressed protein will be a gigantic signal compared to the major peptide from a protein that's expressed at a low level. So this creates trouble, and if we don't deal with this properly, then we will have trouble eating or identifying a proteome. So this is just a comment which I'll come back to just in a few minutes. And I think this is fundamental. This is a fundamental problem or fundamental issue for proteomics as we've been doing it. Okay, so then we are, of course, we all know that some of the issues which you will encounter also in this course, which are common to any bottom-up proteomic methods, we lose the connectivity between peptides and the protein. We make statements about the peptides in one way or another by DDA or by targeting, and then we somehow need to reassemble these identified peptides back into proteins. And the main point I would like to make here, there is no firm rule how these peptides are well, there's no unique way of reassembling these peptides into proteins. All these, this step is usually referred to as protein inference. It is a very tricky business. It is very tricky for any technique, and there is really no true solution These protein inferences all depend on a model, on certain assumptions, and if these assumptions are altered, other sets of proteins are identified from exactly the same set of correctly identified peptides. In the DIA methods, which are inherently targeting methods, we have some flexibility in terms of selecting which peptides we want to sequence. And we can select preferentially, although not exclusively, peptides where the relationship between the peptide and the protein is unique, and we refer to these peptides as prototypic peptides, so they would be peptides which only have one occurrence in the proteome. So if we don't have this, if we have DDA data where we have basically sequenced whatever the mass spectrometer tells us, we have many peptides, like this one here, that may occur in more than one protein, and then the reassembly of these proteins sequences becomes relatively complicated. Okay, so we now use terms like shotgun or discovery proteomics or targeted already, and I think everyone kind of understands these principles, and they will be also explained in the following by Tina, and I don't want to spend a lot of time on that. But in the DDA method, we randomly select, or based on some metric of the mass spectrometer, peptides for sequencing which are identified by the precursor ion signal. We generally assume that what we select here for fragmentation is a single precursor peptide, and if this is not true, if there is multiple peptides concurrently selected, then we have difficulty assigning the sequence. We would now assume that if there is a very large number of peptides in here, the mass spectrometer does not have time to sequence every peptide, simply because if it's overwhelmed by the number of peptides for sequencing. And then it will select the peptides that it thinks it should select, and this has been, of course, highly sophisticated in modern instruments, but it also has, as I'm coming back in a few seconds, led to some issues about reproducibility of the data sets generated by this technique. Targeting does the opposite, or not the opposite, but a complement, and that is you basically tell the mass spectrometer to sequence a specific peptide throughout the whole chromatographic elution time. Basically specify a list of peptides the mass spectrometer should select and fragment, and then you wouldn't generate necessarily a whole fragment ion spectrum, but you would generate traces which are referred to as transitions that elute for a particular peptide over chromatographic time, and the currency of data for the identification of a peptide by targeting is not a spectrum, but it is an assembly, a group of peaks that chromatographically precisely coelute because they're all derived from the same peptide. They have a specific, each peak has, or each ion representing a peak has a specific mass, and which we know what this mass is. It's idiotypic for a peptide. And there is chromatographic parameters like the relative intensity of these, which are nicely conserved. So we're not trying to make a spectrum. We're trying to make a peak group of coeluting transitions or fragment ions of the specific peptide. And so this is a fundamental transformation from going from a spectrum-centric to basically a peptide-centric analysis. Now, neither of these techniques is perfect. We can write down some important parameters: consistency, speed, sensitivity, quantitative accuracy, and each technique has its role, and each one has its specific profiles. If we want to generate tens or hundreds of conditions by this DDA technique, then we run into issues about quantitative accuracy and consistency, and these are nicely controlled in the targeting technique. But the traditional targeting technique, selected reaction monitoring, or also called multiple reaction monitoring, was limited to relatively few peptides that could be quantified per run. So we had two techniques, one which is kind of leaky but goes very deep. The other goes very shallow but is highly consistent and quantitative. And of course the idea would be that would be that we would come up with a method that can go deep into the proteome, but at the data quality of SRM. And so the idea that basically is underlying this DIA method is again borrowed from genomics, and genomics achieved the extremely high throughput now, and also the good accuracy, quantitative accuracy from, for instance, transcript sequencing by going parallel. So this is a picture which probably everyone also knows. This is next generation sequencing, and it is highly parallel in space. So while each nucleotide that is being sequenced is actually quite slow because it has to be generated by a number of biochemical reactions, the whole process becomes fantastically fast by the fact that millions of sequences are being generated in parallel of DNA segments. And how this is being done, I don't want to explain, but I'm sure everyone kind of knows this is basically a parallel in space sequencing where each dot here represents a sequencing template, basically a segment of DNA to be sequenced. And then there's a color-based readout where after each cycle of actual synthesis along a template, we can see which base was added. And we can do this, or the machines do that, by millions of sequences at a time. That's why it makes it so high throughput. So we thought there's the underlying idea of this DIA data is to also come up in a way to sequence, but in our case, proteomes, more specifically digested proteomes, in a parallel manner. But no one knows how to do this parallel in space, at least not now, and maybe someone will come up with this. But it is feasible to do this in parallel in time. So this is a picture that we would basically say represents a proteome of a sample. And this is a retention time dimension, chromatographic retention time as the peptides are being separated. And this would be the precursor mass of these peptides, or more specifically mass to charge. And then each dot here in this window represents a specific peptide that's being generated from the proteins that are being digested by, for instance, trypsin. So we can see here that there's some nicely resolved peptides, but there's also an area here which is very, very densely packed. And there's probably hundreds of peptides overlaying each other. The resolution simply is not good enough to resolve each one. And there's many, and we don't know how many, but maybe there is something in the millions, multiple millions of peptides are being generated from a proteome. We don't know. No one actually knows how many, but it's a very large number. And if we extrapolate from the numbers of peptides that Paola Picotti found per protein, and we then also factor in some modified peptides, the number is very, very large. So the idea was simply to no longer isolate a specific peptide precursor, but to make segments of windows, so basically isolate a number of peptides in a particular window at a time, not specific precursors, but segments, and fragment everything that's in this window. And this is essentially the essence of this DIA method. We generate a certain number of swaths or sections throughout here, and they're seemingly seamlessly annealed with each other. So 32 is not a fixed number, of course. This is simply an early implementation. The main point is that we basically pixelize this whole window here, and that every peptide must be in a pixel, and that each pixel is concurrently fragmented. Everything in one pixel is concurrently fragmented, generating composite fragment ion spectra which are recorded in the mass spectrometer. So these are not searchable by conventional sequence database engines because they are really composed of fragments from multiple precursors, and conventional database search engines choke on those. But I hope that the main message that I'm trying to convey here is that by going in this parallel in time sequencing, every peptide is in principle fragmented in each sample. So the issue of missing values should be reduced or vanishing, and that this can be done quite fast in a single operation. So now, of course, you will talk a lot about how this data are actually analyzed. I'm not spending time on this. I just wanted to say how this method was arrived at. But the main, maybe the only hint already to the data analysis issue is that the data generated here by this technique are structured exactly in the same way as in an SRM experiment. So the techniques that were developed to analyze SRM data, these peak groups, to find these peak groups, to use the parameters that define such a peak group, like the precursor ion mass, the co-elution of transition signals over time, the relative intensity, and other parameters which define this peak group can be identically used to search these DIA data sets in a conclusive manner. So you will learn these techniques, but I think it's good to remember always that the data structure that comes out and the way this data is being searched is exactly conceptually the same as DDA, as SRM data, except on a massively increased scale in terms of numbers. So what's the upshot of this? This is a paper not from us. This is from a graphic from actually from Biognosis, from Lukas Reiter's shop, and he will tell you about the software they develop. And this is now showing the effect of using a targeting, massively parallel in time targeting technique compared to the to a discovery technique. The experiment was that a cell line, a lysate of a cell line was digested. Exactly the same sample was injected in repeatedly, like 25 times, into the same instrument. Same column, same sample, same instrument. The only variable on this experiment is how the instrument was operated. In one way, it was operated in DDA mode, and in another way, it was operated in DIA mode. And then Lukas and his team searched this data and compiled it in these following graphs. So here we see the number of peptides identified per sample. And so this would be in 1 out of 1. We're sorry about the technical glitch, but now it's fixed, but not completely fixed. The aspect is not right, but anyway, we thought we'll continue anyway. And then now the benefit of this interruption was that you could kind of connect with your fellow students. Okay, so I'm going to go another like 25 to 30 minutes. So this is what we were discussing when the beamer went blue. We said the same sample was analyzed in the same instrument setup, and the only variable is the mode of operation of the instrument. And the plot here is, on the top, is the number of peptides identified per run, in the first run, and then cumulatively over the runs. And we see that in the DDA data, we have a certain number of peptides, a high number of peptides, and then when you ask how many peptides are seen consistently in 2 out of 2, 3 out of 3, 12 out of 12, and so numbers and how often are they seen consistently through the data set, which is 24 or 25 runs. The number goes down, and this is what everyone sees, and this is reflected here in the leakiness of the point. So you see in each one a large number of peptides in each analysis, but not every peptide is consistently seen in every run, and the peptides which are seen consistently in every run are usually the ones which are highly expressed. The situation is quite different in DIA, which they call hyperreaction monitoring. So this is exactly the same as what we call SWATH mass spectrometry, simply a different name for copyright reasons. And they see about close to 30,000 peptides per run, and this number of peptides are fairly consistently seen throughout. And this is reflected here in this picture that has relatively few missing values. So if we have the objective or the goal to generate a data matrix where samples numbers are proteins are measured in large number of samples, this technique clearly has advantages, and it comes largely from the mode of operation, not the sample, not the instrument. It also has a tighter CV. So to summarize this part, what I wanted to show is we had stated the goal, and I argued why this is important, that we would like to have hundreds of conditions and certain number of proteins consistently quantified, and this DIA method can achieve that. Although, of course, as you will see throughout the course, there is a lot of room for improvement to go deeper down into the proteome. Now, I would like to spend a few minutes and discuss which dimension of this data matrix, sample versus protein, is more important for cohort studies. So we see that this is a real issue. When papers are submitted and reviewed, we frequently get the response back that with DDA analysis you will get a higher number of proteins. So reviewers criticize that the number of proteins quantified in a sample is not sufficient to draw biological conclusions. So now I would like to comment on this for a few minutes. This is a serious issue, and we've taken reasonably big effort to try to address this, whether this criticism is actually valid. There's no doubt that we would like to, of course, identify and quantify every protein, but this is currently with the technique not possible. So the question is, where should we put our effort to increase the amount of biological information? Is it in the dimension of more samples, or is it in the dimension of more proteins per sample? So first, I use this slide to set roughly the scene, what we know about the proteome. So this is work from Martin Beck when he was a postdoc here, and he tried to estimate the quantity, the absolute quantity, the concentration of proteins in a human cell line, and this is what he came up with. So I don't describe how he did it, and the values are not super precise. They're probably precise within a factor of 2, but the picture will remain the same even if the numbers are not super precise. So he basically made a list of proteins and estimated the copy numbers per cell. So what he concluded is that, or what the data show, is that if we identify, we shouldn't talk about identification, we should ask what fraction of the proteome is represented by a certain number of proteins if we list the proteins from the highest expressed to the lowest expressed as they were detected. So the data suggest that the 500 top highest expressed proteins would cover about 87% of the total proteome. 1,000 proteins, 94, 1,500, 98, once we're up to 25 to 3,000 proteins, certainly 99% plus of the total proteome mass is covered. So now what does that mean? I would like to come back now to the mindset of proteomic studies. If you have the mindset that you need to find a specific factor that is carrying out a specific function, let's say a transcription factor, and you would like to detect this transcription factor for biochemical reasons, then, and this transcription factor is not in these 3,000 proteins, then of course you will get no result. It's a dead end. You need to dig deeper. If, however, the question is, how does the proteome react to a specific perturbation, let's say the absence of a protein or a mutation, then we can assume that if this protein, even if it's a low abundance protein, is knocked out, that it will very likely leave a measurable footprint in the proteome that's measurable in the 99+% total protein mass. It is inconceivable to me that a protein would leave a footprint that in the proteome that is only present in the, let's say, 0.5% of the proteins which are not of protein mass that is not visible by a technique. So typically now, Initially, you will see this also through the exercises. Initially, this is roughly the number of proteins which came out of DIA data. Now, with better instruments, this has gone out to from cell lines to 5,000 to 6,000 proteins. So this also, of course, instrumentation has improved a lot, and the coverage has become also higher, but still, of course, not every protein is covered. Okay, now this is work. We wanted now to, within this framework, we wanted to explore which dimension of this data analysis, of the SWATH data, number of samples versus number of proteins identified, where should we put our effort? So the mindset would be that we have a laboratory, we have certain resources to do a certain number of LCM SMS runs. We would like to do a certain type of study. This is modeled around a biomarker study. And the question would be, where do we invest our resources? Should we try to run more samples at the price of being somewhat more shallow, or should we go deeper into the proteome, which of course we can do by fractionation? So this is work from Ting Wan, who is a PhD student, statistics PhD student in the lab of Olga Witek, and she has generated a model, a model in a computer that allows us to make simulations. So this is the question: should we, if we want to do in the context of a biomarker scenario where we have two groups, let's say healthy control and disease, for instance, cancer tissue, and we assume that there are proteins in these respective proteomes that distinguish these two groups, which is a classical question of biomarker analysis, where should we put our effort in having more subject or more proteins? Okay, so what she did is she took 3 data sets which are published, generated by different methods. The one is by DDA spectral counting, one is by SRM, and one is by SWATH-MS, and she learned basically from these data sets the noise behavior of the data. With noise behavior, I mean is how variable, what's the technical variability of a protein measurement as a function of abundance. Every analytical technique will show something like this, which is usually referred to as a trumpet plot. And this simply means there is a plot of the signal intensity, high being over here, low being over here, and then the standard deviation of measurement, basically the technical noise. And every technique is doing reasonably good at high intensity signals. And when you can come with the signal down to signal to noise level, then of course it gets more noisy. So this is what one would expect here, and this was the learned behavior of the SWATH data. This is the behavior of the SRM data, and we see that this does not show this trumpet plot, and the simple reason is that these proteins were selected by SRM to be proteins which are very precisely measurable. So we never even got into the noisy range here in this data set. And this is the data set just generated by the spectral counting, by the CPTAC consortium. And this has an entirely unexplainable behavior, being that it also is reasonably tight. It goes up like a normal trumpet plot, but then it goes down. And this behavior is unexplainable. We called them and we wrote to the authors of this paper, and no one can really say what they did or they don't want to say. So we don't know why it goes down, but this is clearly an artifact. There's no analytical method known that would become more precise the smaller the signal gets. So this has somehow to do with data management or with data analysis or data processing, so we should simply ignore this branch, but here it behaves normal. Okay, so you see here also there's different numbers of proteins that were analyzed in this data set. So Ting wrote a computer program that basically learns this behavior from the different data sets, and then it allows her to set up a specific scenario. She basically takes these proteins that are observed in each one of the data sets with the quantitative behavior, the noise behavior, the technical noise, and she makes 5 quintiles, quintile 1, 2, 3, 4, 5, and then she puts in a simulation in each one of these quintiles biomarkers, proteins, signals that would distinguish these groups. So now we can say, if we had under certain behavior of technical noise, which is inherent in the respective methods, if there were 3 predictive biomarkers that separate the groups in this highest group of proteins, or in the highest group, or in all groups, including the more noisy proteins, then what would be the performance of being able to separate the healthy and the healthy control and the disease? And she can do this, of course, by simulating the number of proteins, by simulating the number of samples in a computer. So this is the result, or this is the design. It simulates training and validation set with different numbers of proteins and subjects, using the realistic backdrop of the noise behavior of the data sets as they were actually measured. And then she trained the system and makes predictions of performance. So the groups of number of proteins she tested was 60, 500, 1,000, 3,000, 5,000. Biological replicates 10, 40, 100, 200, 400. And then she had various distributions of the biomarkers in this data set. And then she does this many, many times over. Okay, so what's the result? The result is, the essence is, every time, in every simulation, the number of samples wins over the number of proteins. So the conclusion is, I'm coming to what the data show here, but the conclusion at the end is that if we have a certain number of runs or measurement capacity in the lab, and if we want to do a study that depends on correlative analysis or biomarker of the biomarker type or otherwise, every time we win if we have more samples, even if it comes at the cost of the number of proteins. So how do we see that? I mean, if we see interesting behavior, this is the number of subjects, this is the number of proteins. We see that 1 is high, and the lower the number, the worse is the performance. So here's, for instance, the ability, the sensitivity of identifying predictive proteins, basically the ability to separate the groups. We see here that with the increasing numbers, the color gets more and more violet, so it comes up to blue, to 1, which is the optimal performance. So this is a very clear trend, and in every simulation that was borne out. There's an interesting observation here with the number of proteins, because we see that the more proteins we add in this predictive accuracy, but also in the ability to separate, the performance actually goes down. If we have, in every case, so we have here with relatively low number of proteins, we have good separation, and then it starts gradually going down the more proteins we add. This is counterintuitive, because if you go to a proteomics laboratory, you would mostly assume that most people would say the more proteins you add to your data set, the better you're going to perform in a biomarker study. And the data clearly show that this is not the case. So why is this not the case? The reason is because when you keep adding proteins, by necessity how the proteome is constructed, you end up with less and less abundant proteins. You get more and more into the low intensity domain, and then the data become more noisy. So we can see this from this curve here. When you were adding low abundance proteins coming from this side here where the coefficient of variation is low, and then you get into the realm where the coefficient of variance is rather large, so the technical variability is rather large, then you start to drone out, simply because you generate noise in your data set, you start to drone out signals which were detectable before, when it was less noisy. So the conclusion clearly is that every time, if we have the ability to either go for more proteins, let's say through fractionation, or if we have the ability to go to larger data sets, in a data-driven approach, we should always go to larger data sets. Now, this may not be possible because maybe in a clinical case, we might only get relatively few samples. But if the option exists, the data always wins. Secondly, we should focus not on the number of proteins, but on the accuracy of measurement. So what we would like to achieve, I think this is a clear analytical challenge, that we would like to move down this trumpet plot, the behavior of it, so that we can stay longer in a domain of less noisy data. And so we should try to avoid noise, technical noise, and not necessarily try to get at every price at a higher number of proteins. So this is the conclusions from this. More proteins, decreased predictive accuracy and sensitivity of identifying predictive proteins simply because it becomes more noisy. So we have to pay attention to noise, not so much to numbers. More subjects, increased accuracy and sensitivity even within the same noise regimen. And less technical variability increases the performance for sure. And I think this is an analytical goal to, now that we understand this behavior, to reduce the noise for, or increase the signal, or increase the signal to noise for lower abundance signals. So the conclusion would be that the experimental techniques should optimize for throughput of biological samples, low technical variability, and high reproducibility, as opposed to making identifying longer list of proteins. So now as we're talking about variability, I would like to now, I'm not going to talk about this, so just we clearly now established that variability is important. And on Friday, when I have another talk, I'll come back to this issue from a biological point of view. And I now want to make a few comments about the question, can we quantify sources of variation, and why is this important? So we normally assume that, that what we measure in a specific biological situation is representative of the underlying biology. What we usually do not factor in is that the underlying biology inherently is noisy. Now, not just talking about biological noise, and not just technical noise, as we just discussed, but also biological noise or variability. And it is important to know, for reasons I'm going to explain, how this biological noise, whether it exists, whether we can detect it in clinical noise, in technical noise, and what we can learn from this, from the noise behavior, the variability. So let me use an example here from a study that a postdoc, Yansheng Lu, carried out in the lab with DIA data a while back. So there is hundreds of thousands of papers have been published about protein plasma biomarkers, and many of you may actually also work in this field. So the idea would be exactly as we discussed before, just a minute ago, to have two or several groups, and we would like to find proteins in these groups, let's say healthy control and disease, that separate these groups apart. So lots of papers have been published, and we don't know the basics. So we don't know the variability of protein abundance in a population. Biomarker studies are always carried out in a population. We don't know the source of variability. We don't know the variability as a function of time. So let's say if you go to the hospital and get 50 samples from cancer patients of a certain type and 50 control groups, and when we want to do measurements, we don't know any of these factors. And I now show that these factors actually are highly important for the interpretation and the success of such biomarker studies. So why am I saying that? So let's say we want to measure a plasma protein that can separate two groups. Very typical question. And let's say the ideal case would be that this would be the one group, this would be the other group, and the measurements in one group, let's say the healthy, would be very tight. The distribution of abundances in the case group would be also very tight, and they would be significantly different. Then we have a good chance to actually find a biomarker and say we have a case where we can separate these two groups. If this is more relaxed or variable, and this is more variable, the two distributions start to overlap. And if it's even more variable, then we have essentially no chance of finding a biomarker. So typically, the question that is being investigated, how far apart is the mean of the one group to the mean of the other group? And oftentimes, the variability here within one group is not really factored in. And apart from the technical variability, which of course is inherent in every technique, which we just discussed, there's also biological variability, and this is what we wanted to, Yansheng wanted to explore. This is a case of a protein where over a number of cases, a number of individuals in a population, the level is fairly tightly controlled. This is a case of a protein where the level of the protein in a population is very poorly controlled, or it's very sloppy. Shouldn't say it's poorly controlled, but it's much more variable, not for technical but for biological reasons. Now, if you wanted to use this protein here and develop into a biomarker, then you would probably be in this or in this domain, and you would simply fail. And so we think that many of the reasons that many biomarker studies fail is because the behavior, the variability behavior in a population is not really well understood. So Yansheng Lu did the following experiment. He generated or recruited a twin study, a population of twins, some of which were monozygotic twins and some are dizygotic twins. And then he measured quantified by SWATH DIA plasma proteins. So why a twin study? Because a twin study allows us to assign the source of variability to either genetic reasons or non-genetic reasons like the environment. The reason for that is monozygotic twins are expected to be genetically essentially identical, and if the variability is different from monozygotic twins on dizygotic twins, we can calculate the difference in variability in the same cohort and then say if the monozygotic twins are tighter, the variability is less, that the abundance of this protein has a component that is genetic, a genetic component that determines the abundance and the variability. So formalism has been worked out in this. This is the cohort. There were a number of monozygotic twins, a number of dizygotic twins, and they were all sampled in 2 time points about 5 to 7 years apart, and then the proteins were measured. So this is the layout: 36 monozygotic twin pairs, 72 individuals, 44 dizygotic individuals, measured twice and measured in duplicate. So this is a fairly high number, about 200 plus SWATH maps were generated here from plasma. This is the data. This is the showing that the variability that we see can be distinguished. The biological variability can be distinguished from technical variability. This would be the 6 technical replica. So they are, and this is basically the coefficient of variation measured for each protein. We can see that the blue curve is considerably tighter or considerably has less variability than the red curve. And the difference between the blue and the red curve, we assume, is since the same measurement was done, the same sample workup, so we assume that this is biological variability. So what this means is that even though the technique is of course not perfect, some proteins have a certain degree of technical variability, again reflecting this trumpet plot situation. By lower abundance, you get more noise. We can clearly see distinguish technical variability from biological, and therefore we can see biological signal. That's just to state the obvious. We also see that various proteins have very different variabilities, actually on the level of peptides, but that the variability in this cohort of individual peptides varies greatly. So, and this is not just for technical reasons, it's mostly also for biological reasons, as I just showed in the previous curve. We have a number of peptides or proteins by implication that are quite tightly controlled in a population, so the proteins tend to be relatively have low variability, and some have relatively high variability. Now, this formalism that's simply an observation, is not super interesting. Now we can try to find out, using this formalism that is inherent in these twin studies, where does the variability come from? Is it genetic in origin? Is it from the environment, or is it age, or some other factor? And this is formalism that was developed to do such studies, and we basically can observe the total biological variability within this cohort for each protein, and can we attempt, using this formalism, to split apart the variability observed into genetic reason, into environmental reason, or other factors. So this is a summary of this data. So this would be the proteins that were identified, and this would be the percentage of variance, that contribution from various sources. So the red one is heritability. So we see that there is a number of proteins in this cohort. And I should see, this is not a very deep proteomic study of the proteome. The number of proteins actually quantified was about 400. Of course, this is only very shallow, and one would like to go very deep into the plasma proteome, but it's technically very difficult. But within these proteins that were found and quantified, there's some that have fairly substantial genetic control. So their variability or their abundance level is pretty tightly controlled by genetic means. We don't know, of course, which locus necessarily, but it's simply stating a fact. And then there is proteins where the environment, so the blue is the environment where these individuals live in. There's some that have very high, are highly dependent on the environment, and others, in others, the environment is not really a big factor. And then there's also longitudinal effect. This is basically the age of the person. We see proteins here where the age doesn't really matter so much, and we see proteins over here where the age makes a big difference. So the protein variability, the protein variance, is very different at a younger age and an older age. So these are simply stating the fact. And now what is the implication for biomarker research? So when we looked then, or this is Yan Sheng's work, he went through the literature, and he pulled out some papers which had postulated that certain proteins in these papers were biomarker for a particular disease. So there's hundreds of these papers, and many, of course, postulate biomarkers that were not in this data set because the data set is quite shallow. But there were some, these are these papers, where the authors postulated that they had found biomarkers to distinguish, let's say, case and control, based on plasma proteins they identified. None of these has made it over into the FDA or actually into clinical practice, and I think now we understand why. We see that each one of these proteins has a substantial variability in terms of age. So the yellow is the longitudinal effect, or the age. So what this means is that each one of these proteins, which were postulated here and published as biomarkers for a particular clinical case, were proteins which have where the abundance of these proteins is strongly dependent on the age of the person. So now, so you can immediately see why this is a problem, because when you now were to collect a cohort of patients with this disease, and it might well be that simply what was measured here is an age distribution of, for instance, the control samples or the case samples. And so these proteins that are all here are listed in these papers as biomarkers did not make it into biomarker because there's clearly factors outside of the underlying biology which misled the authors to claim, basically these were confounding factors which showed up here, which were not understood. So we also, this is something I don't have on the slide, Yan Sheng also looked for the noise behavior of proteins which actually made it through from clinically used biomarkers, and some of which also showed up in this data set. And he asked, what's their behavior, where is the variability in a population coming from these proteins. And inevitably, any one of these proteins has a strong genetic component and a very low environmental component. So generally, we can conclude that proteins would make it through to biomarkers, validated biomarkers, if, of course, they indicate some effect, if they reflect somehow the underlying biology of the disease, but also only if they are relatively robust and relatively tight in a normal population. And for most proteins, this is not known. And if we knew these effects, which only can come out from relatively large-scale clinical cohort studies, like, for instance, the one here, then we would be able to weed out most biomarker candidates simply on their behavior of variability in a population, because many simply will never have a chance to be discriminate proteins to discriminate cases and control, as I try to show here in the initial slide. We would need to do some selection if biomarker samples, a biomarker study should be successful, to see whether the data situation is actually like this here, in an ideal case, maybe slightly less ideal than this. So this protein would have, in principle, if it reflected biological information about the disease, would have a good chance to make it into biomarker. This protein will never have a chance to make it into biomarker because it simply is too variable in a population. And depending also even where the sources of variability comes from, if it's simply an age effect or an effect to exposure to certain life conditions, the chances that the protein will make it into biomarkers is even lower. So now, of course, these comments are not just true for biomarker research, it is true for essentially any research that we want to do with big data. We need to make sure that we understand what the variability of the effect is. We need to be able, that we want to study other proteins. We also need to know whether we can distinguish noise, technical noise, from biological signals. And again, I'm coming back to the issue that it is not just the number of proteins covered, but the precision with which these proteins can be measured reproducibly for biomarker studies, as well as any other study that depends on large-scale data sets. So the conclusion from this plasma experiment was that such a data matrix could be generated, and it provides biological and technical insights which are otherwise difficult to get if you focus on a specific protein at a time. The abundance of the plasma protein varies across the population tremendously, actually, and the degree of variability and the sources vary for different proteins. So there's no fixed rules. Of course, the rules are not understood, but it's simply an empirical observation that there's a huge amount of difference in the variability and also the reason why proteins are variable. And highly variable proteins make poor biomarkers. There will also be bad proteins for any other association studies, because when you have a very noisy situation to correlate, then you will of course suffer from in the quality of the correlation. And confounding factors like age will lead to wrong results if not recognized and corrected. So with that, I would like to finish because my time is up. I wanted to actually make some comments about this, but I'm run out of time, and it will probably come up in the course. So what I wanted to show is that every experiment that we do, when we enter this relatively large-scale biology regimen, will have factors, confounding factors that we don't understand. For instance, the machine might not be in the same quality from one day to the other. Sample preparation might slightly vary, might use a different buffer inadvertently. Even if you try to do exactly the same experiment, no one is ever able to do something exactly the same. So since we're now dealing with hundreds of samples in a typical study, it is very important to design the study in such that if there are effects, like I showed here with these age effects in this plasma study, that this would be recognized and then they can to some extent be corrected. If these factors that distort the experiment and the results through an artifactual effect that we don't anticipate and we don't control for in the design how that we can have an experimental design that actually allows us to recognize that there are such batch effects or confounding effects, then we made the wrong conclusions. So I think what we never, I think in the proteomics, certainly we never worried about this, because we're usually analyzing only relatively few samples, but as deeply as possible. Then these issues are not really so difficult. But once we start to do real cohort studies, the control of inadvertent variables becomes very critical. And if you don't try to prevent them or to recognize them and then possibly correct them, then this may lead to wrong conclusions. So I'd like to stop here, and I wish you a great course. I'm sorry for the technical glitch, but it's partly fixed, so he needs to fix the format, but at least we could continue. Thanks for your attention.